Hello, good afternoon, everyone. My name is Lee Ullman, and I'm the Senior Director of the MIT Sloan Latin America office. This office is part of MIT Global Programs. The goal of the Sloan Latin America office is to share MIT knowledge with Latin America and to look for collaborations and partnerships with the regions. As part of the office's role, what we do is we offer seed funds each year to faculty and researchers at MIT to provide them with an opportunity to work with partners in Latin America and also to come visit the region to find new and unique projects. Today, we have one of the awardees of the Sloan Latin America Office Seed Funds, and that is Dr. Mercedes Balcells. Mercedes is Principal Research Scientist at the Institute of Medical Engineering and Science. Her research bridges the cardiovascular and neuroscientific domains. In 2011, Spain's government bestowed Dr. Balcells with the Cross of the Order of the Civil Merit for her contribution to establish a fruitful channel between MIT and her home country. I'm very excited to have her here today and to share this fascinating research with you. In a normal year, we would have Mercedes with us down in Latin America so that we can actually have her cross the equator. Mercedes explained to us that she got as far as Aruba. So she's gone a lot of east and west, but never south. So we're very excited once the pandemic passes to bring Mercedes down to the region. So I'll let Mercedes give more of an introduction, but I did want to thank Jacqueline Taylor from Global Programs for helping set this talk up today. So without further ado, Mercedes, all yours. And one last thing that I'd just like to tell everybody is if you have any questions, please put it in the chat or the question and answer. We'd like to make the talk as interactive as possible. So thank you again for being here today with us.
Thank you very much, Lee, for the introduction. Can you see my screen? Yes? Okay, perfect. So I'm going, since you did such a wonderful introduction about all my achievements, you made me a little bit self-conscious. I would like to start sharing a little bit of more of a personal story. And this is the first slide of a short talk to share with you my work here at MIT. But I come originally from Barcelona, where the architecture is amazing. And I am the daughter of generations and generations of architects. So I'm sort of the black sheep in my family. I'm the only one non-architect. But definitely architecture has influenced my career, and you will know in the next slides where you see some scaffolds similar to those used in architectural renovations. The other thing about myself is that I am a passionate skier. I sometimes forget my age, and so I'm still 15, 16, 17, and I love ski and speed and taking risks. That is also something that happens in my laboratory and my personal life still today. I became a scientist because I had a great science teacher, a chemistry teacher. When I was in 7th grade in Barcelona, the chemistry teacher was trying to teach us the elements of the periodic table. And she did just something, is to mention that the periodic table and what we have in the books is just the tip of the iceberg, so that actually there is much more unknown. And I think that day I became a scientist, an explorer. And I started making very non-orthodox experiments with my big, crazy Spanish family. This is the photo on the right. My mom always wondered why all my siblings, I'm the oldest of 6, by the way, and we spent a lot of time in my grandparents' garden every weekend. And I wanted to investigate what eatable plants and flowers were in the garden. So I will prepare some mixtures and feed them to my younger siblings. And so my mother always wondered why everybody was having some stomach issues except of myself, the doctor, the scientist. So that's a little bit about me. When I have a more formal talk and I start saying that I work on tissue engineering. And a lot of people ask me if I do, I don't know if you can see my face now, if I do tissue paper for living. And it is not exactly tissue, or in Spanish we say tejido. It's not clothes, although clothes are fabulous. But it's this kind of tissue, is organs and tissue substitutes, so that when those tissues and organs fail, we can understand why they fail and how to replace them or restore their functions. So my lab, the main ingredients in my lab are human cells, are scaffolding materials that we use, we synthesize or in the lab, or we use nature-inspired materials. The cells grow on those scaffolding, and very much like in a building, once the cells attach and start producing their own material, the scaffold degrades and disappears. Also, something very important, you can imagine that if you have cells in your body, each cell in your body reacts to their mechanical environment. Think of your knees, you know, the compression that they suffer every time you walk or you run. Think about your arteries and the blood circulating. There is this friction force of the blood shearing against the walls of the arteries that are cells. So I'm very much interested in how the mechanical environment affects human cells in different, as I said, organs and tissues. This has tremendous implications in cardiovascular disease, for example, where we are building replicates of blood vessels, in brain science too. And most recently, since you develop replicates, substitutes, you can test them, and you can test them under pathogens, like, for example, viruses. The project that I presented to the MIT Sloan Latin American office, it's a very new one. It's actually bringing together different dimensions and different disciplines. And we just started in the fall. It's bringing together cellular biology, cellular and molecular biology, understanding how the cells in your brain work and when they don't, and bringing together behavioral science, psychiatrists and psychologists to see if there are better ways to measure and monitor and manage anxiety and depression disorders. And if there is a correlation between anxiety and depression and chronic disease. And instead of using, in addition to using cells, also using something that is perhaps less invasive, although invasive also, and is your cell phone, something that you carry with you all the time. Try to see if we can do some continuous monitoring without you, without invading your privacy, obviously, but without having to ask you to donate some cells from your own body. And this is in a nutshell what the project, the big picture project, is about. Let me clarify or get a little bit more into the science. I hope you enjoy knowing that we started building some years ago with students that I have that are the jewels of the crown. We started building MOCs, replicates of arteries that irrigate your heart, carotids, coronaries and carotids. They irrigate the heart and the brain. And with this, as you see the picture on the right, with this one-to-one scale replicates of somebody's arteries, we would start to analyze and do some computational analysis to know how blood flow affects those arteries. These are not discussed, these molds were then layer by layer with the different components, cellular components that make an artery, that are adventitial fibroblasts, aortic smooth muscle cells, and endothelial cells. You see them here in this beautiful picture, the different cell layers stained to distinguish them so that they assemble layer by layer inside these geometries. Well, we also had to answer a lot of questions because sometimes you put cells layer by layer, that doesn't mean they act as an organ. So we were testing a lot of functionality like vasodilation, vasoconstriction, clotting ability, what happens if you injure the artery, what happens, for example, if you introduce a vascular stent because the artery has been occluded. A bunch of questions that these artificial vessels became excellent models to test. And we test them, as I mentioned, the mechanical environment is critical. So if you come one day to my lab to visit, you will see a lot of machines, instruments that we have developed. Sometimes I think they are training machines for the cells, like a gym for the cells. Sometimes I think they are torture machines for the cells. Some of my students, I come into the lab and say, all the cells died today, Dr. Balcells. I'm like, oh no. In a nutshell, what these instruments serve is to expose the cells on those tubular structures to the mechanical environment that they will be exposed to if they would be inside your body. And you can, with the advantage that this is not a person's body, this is just layer by layer assembly of human cells in different geometries, so that you can test the system and see push, push, push. Like, for example, imagine you can increase the pressure until they burst. You can stop flow and see what happens with stop stagnation, flow stagnation, and so forth. You can torque, you can bend, you can pull, you can shear, you can really apply a bunch of parameters to the cells in the constructs. More recently, our research focused on understanding a very special type of blood vessels, and these are the capillary in the brain. Those capillaries have a layer inside of endothelial cells that are very particular because they create tight junctions that preserves the brain intact. And then the part outside of the capillary that are astrocytes and neurons. So this connection between these two types of cells build what we call the blood-brain barrier. And we have been able to recapitulate the blood-brain barrier. It's a barrier, and that's why Ramon y Cajal, the Nobel laureate from my country, received it, discovered more than 100 years ago that the blood-brain barrier protects the brain, so that if you inject a dye, for example, in your vein, all your organs will turn, if the dye is blue, all your organs will stain blue except of the brain, because the brain is isolated and protected. And that's a good thing, you know, and that's why it's so scary when something happens in the brain, because that thing, for example, meningitis, that pathogen should not be in the brain because the brain is protected. And if you want to treat it, how can you permeate a barrier? So it poses, it protects us, right, so that we are not extinct, but also it prevents treatments. So in the lab, we have developed a model of the blood-brain barrier. I have very smart student, Gonzalo, dealing with different materials that imitate the pore size and the elastic modulus of the brain. Here on the bottom, you can see different stiffness of this material done with biological molecules that can be cross-linked and reacted to make a more tighted net or looser net. So basically playing with the mechanical parameters that are possible. And this is very important because this material has to have certain stiffness in the interior, so that the astrocytes is this photo on the right that looks like a star, is the brain astrocyte. This is a happy astrocyte. It looks like a star, it's feeling good. If you put the matrix too stiff, you would see the same cell with all the legs and the projections curling up and becoming circular. That's an unhappy one. On the other side, on the top of the inside of the capillaries exposed to blood flow, you have the endothelial cells that look exactly like this. On the left, you can see in green what they are the junctions, the connections between every cell. And those cells are extremely mechanosensitive, and they need a stiffer matrix underneath them. If it's too soft, they will peel off and the blood flow will remove them. So it was a lot of work to get this model put together. In addition, we connected it, as I mentioned before, with the bigger arteries to flow, because the barrier will not be fully functional on a stagnant, no flow condition. So we developed the device to be able to impose flows that are typical from blood flow in the brain. These are, again, beautiful pictures to show that in our model, the cells align. In this picture, it will be like diagonally, the blood flows so that the cells align in the direction of the flow. The color green and red show the red is ZO1, which is a typical one of the characteristic proteins in the tight junctions. CD31 is one marker for endothelial cells. So you can see the cells at great detail and see how they are doing because the device is transparent. And one thing we test is what kind of shear stress is beneficial to the cells and when that shear stress is no longer, is harming the cells. So in these 8 pictures, in the center top B and C panels, you can see again the endothelial cells elongated right to left in the direction of the cells, in the direction of the flow. And these are 10 and 20 dynes per square centimeter, which is the average shear stress levels in the brain, in a healthy brain. Steady flow, meaning constant. There is no pulsatility in the brain. The pulsatility of blood flow provoked by the heart has been damped when the blood reaches the capillaries of the brain. So this would be the healthy state of the cells on B and C. If you add pulsatility, you can see that the stain, the green color goes into the center of the cells, and you lose the junctions. The limit between the cells becomes blurry and actually much less intense. If you stop flow on the left column, both, you're still seeing serrated patterns on the periphery of the cells, on the membrane of the cells, and that serrated pattern suggests disruptions of the blood-brain barrier. And on the right, if you impose a very strong shear stress equivalent to having a high pressure, again, you lose the resolution of the tight junctions that ensure that the blood-brain barrier is working properly. Not only have we looked at how the cells look, we are because obviously if I ask you, can I look into your cells in your brain? You would be like, whoa, Mercedes, leave my brain alone with this, because all these have been done in cells isolated and in models in the lab. So the idea would be, can we harness something that the cells produce themselves and that we could analyze and detect and characterize without having to open anybody's skull? And the answer is yes. The blood-brain barrier is, as I said, is a very tight barrier. Only small things can go across it, small molecules, and most of them are from the out from the blood vessel in, and they are mediated by certain molecules that are outside of the cell. They are receptor mediated, like insulin, carrier mediated, like glucose, small amino acids, but there is something that the cells produce, any cell in your body, not only the brain cells, but for sure the brain cells, they produce nanovesicles, and those are endogenous. They are little vesicles made of membrane, either nuclear membrane or cytoplasmic membrane, that the cells expel out of their inner. And if we could isolate and characterize those nanovesicles, those would be in your bloodstream. And there is a lot of publications suggesting that those nanovesicles can be markers, biomarkers of certain diseases. So we have been I think sometimes an image is better than many words. These pictures, are the nanovesicles that we have been isolating. And I call them vesicles, nano and micron, because they have different sizes. Just to give you a comparison, in the second picture on the right, you see this round, I am marking here, the round black circle. This is the pore of a filter paper. This pore is 0.2 microns in diameter. So this is 0.2 microns, and these are the vesicles we are isolating, which are like 1,000,000 times smaller than a hair. And we are able to isolate them by different methods. Some are really tiny, like the picture on the left. Some are a little larger, like the second to the right, where they aggregate, and then they become more and more complex. We don't know, we are still in the work to characterize those vesicles and understanding why they are so different, and are they produced differently if you are healthy or you are stressed. If you are under stress, and then if you have been diagnosed with anxiety and depression or other disease. So these are the vesicles that we are working on in the lab right now. But let me bring you back now to the microscopic world before I get too deep into the science of nano. When you go to the doctor, many of you, I certainly have, you go to your physical and you answer certain questions before you even see the doctor. These are standard questionnaires. One of them is called PHQ-9, that are to diagnose anxiety and depression. And based on the score that you give on this test, and if you answer a certain number of these questions with a high frequency, nearly every day, you would be diagnosed. The same happens with chronic disease, like cardiovascular disease. You go to see your doctor, and at discrete time point, when you are in the doctor's office, they will measure your blood pressure, they will listen to your heartbeat, they will take your temperature, discrete in time, and these are classic measurements used by the doctors right now. The question now, this project that we have started is going through dimensions from the nano and gene experimentation all the way to see if we can include other type of markers, not just the questionnaires, not just your doctor, your cardiologist looking at your heart and measuring your blood pressure, but seeing if it's possible that we can do something that can be more continuous monitor. And if there are other markers that we can use, for example, not only what I mentioned before, take a simple blood sample or a urine sample and look at the vesicles, secreted vesicles, and be able to relate them to a disease or a condition, but also using your cell phone. The cell phone, your cell phone and other wearables, with or without other wearables, is able to collect up many parameters, 24, 30 different parameters. It detects light. It detects where you are, geolocation. It has an accelerometer, you know, how fast you are moving. That's why you can download an app that tells you how many steps have you made. And all this is collected without you realizing. And all this data could be used clinically to see if what you score when you are discrete in the doctor and what your phone is recording continuously, if there is a correlation. That would allow for early intervention and prevent, you know, the disease to go further before intervention. The collaboration is key. This is one of my last slides. Going from gene to behavioral and intervention, using computer science, using sensors, using biological markers of 10 to the -9 dimension, using and dealing with all the spectrum requires a lot of people. I'm blessed to have, you know, colleagues, Dr. Edelman, Dr. Goss, Dr. Lapedriza at MIT, a wonderful team of undergraduates, students here at IMSS, that we are working on the sensors, biological and digital. We have established a collaboration not even a year ago with Albizu University in Miami-Dade County, with a group of clinicians that are experts on mental health disorders, and they are clinical psychologists. Just recently, I got the good news that I have been awarded a Misty Mexico Fund to collaborate with an alum of MIT, Dr. Adolfo Guzman, who graduated in CSEL some years ago. And they are experts in computer science. So they are going to be analyzing the data that we will collect. And thanks to the MIT Sloan Latin American office, I have been able to reconnect with a longtime collaborator, Dr. Jose Krieger at the Heart Institute in Sao Paulo, and two alums in my lab, Dr. Miyakawa and Dr. Dreyfus, who are in the Heart Institute as well. So this is a big collaboration with different, as you see, different specialties, from cardiology to psychology to AI to sensors. But I think this is what is, I strongly believe that this is what is required to manage what is I think the epidemic and the pandemic of the 21st century, which is not COVID, is mental health issues. there is a correlation I didn't mention before, and a lot of studies suggest that if you have chronic disease like cardiovascular disease, you end up developing depression in many, many instances, more than 50% of the patients, and vice versa. If you are diagnosed with anxiety and depression, you end up developing cardiovascular disease because your habits of health habits, you start eating poorly, not exercising, maybe drinking too much. So there is a tremendous overlap between these two, and this collaboration, we hope to bring some light on ways to get them early from different angles, disciplines, and to be able to keep people healthy longer and without having to go to the doctor. As I said, going into the doctor is a single data point where it's not where you are in your normal life. And I know this slide is busy, but I want to acknowledge that this is not research that has been done overnight. It takes time, a lot of people, tremendous students, funding from different companies that supported my research, but also from the Ministry of Economy of Spain, and a lot of wonderful colleagues here in Boston, in Spain, and everywhere. And I think with that, I leave the floor for questions and whatever you want to ask me. I'm here also. I was very happy when I got the award and I was invited to the webinar, because I thought it would be a good opportunity to see if I can start new collaborations with some of you in the audience, and if you have any recommendation, because as Lee mentioned, I have never been able to visit and to engage in person with partners in LATAM. And I am a mom of 2 girls, and every time I fly, it is to Barcelona, Boston, Boston, Barcelona. My flying has been between the business of the lab and my big Spanish family. My travel has been limited or reduced to go to Barcelona. But now that my girls are getting older and the pandemic is hopefully going to end soon, I would be delighted to visit.
So thank you so much, Mercedes. There is a question from the audience, and then I'm going to ask you some questions. So the first question comes from Andres, and he said, can the blood-brain barrier be expanded, contracted to let smaller, bigger molecules through?
It's a very good question. And actually, the quick answer is yes. You can apply a stimuli that creates nanopores, very transient. Like for example, ultrasound. You give a pulse of ultrasound and you can expand, open certain pores to get something in, and then the pore will turn. The problem is in clinical practice is not applicable because of the ultrasound not getting there. It's too far off. But in the lab, we are looking at ways like non-invasive ways to non-permanently open the blood-brain barrier. That's for therapy. In terms of for disease, it does get destroyed, and that's the problem. When I keep telling my daughters, you take one drug, one chemical drug, for recreational drug, and that drug can damage your blood-brain barrier for life, one single dose. So it gets, and then it gets leaky, and that's the problem. It gets leaky. It's supposed to be a barrier. So do not use drugs.
It's a great message. But let me ask you about that, because just to go on that line, again, some of the people in the audience are non-scientists. So we all know if you have depression or anxiety, the pharmaceutical companies have tons of different medications and different therapies for people. But if I understand correctly, what you're trying to do is look at a new way to monitor and see other ways so that at least medication or these potent drugs are not the way to go about it. So is that a good way to see it? Or how would you, as a scientist in your lab and not a Sloan business person, explain it?
Yeah, you have made a good point. The idea is to prevent disease, so that you don't have to treat people, you have to prevent them getting sick. And yeah, before pharmaceutical intervention, the ideal will be to be able to diagnose early and have some treatment that it doesn't require, a big drug or a big pharmaceutical formulation having to go through your brain. So the idea is early intervention. That's why we are working with clinical psychologists to, through the phone, not only measure those 24 parameters that I talk about, and they are studies that are fascinating. They are not for for mental health, but for example, monitoring students in campuses and looking where they are, if they are in the library studying, if they are in a place like a restaurant or socializing, and see how that has an effect on their grades. So sociability and grades. So one can think of similar tools to see how you are doing, with your daily life, prompting some questions of those questionnaires that the clinicians use once every 3 days and saying, how are you feeling? Are you feeling sad? And then relate of where you were and be able to see before it's too late that that person's mental health is deteriorating and being able to intervene. I have not mentioned, but we are also starting to work on ways to be able to intervene early.
So Mercedes, another question is how do you visualize your research ultimately to apply or to diagnose treatment for Alzheimer's? So is there other aspects of your work that would, I guess, treat other diseases or chronic diseases because of this brain barrier? Or how do you see some of what your work can be applied to?
Yeah, as I mentioned, my mini brains, the blood-brain barrier substitute is just a model, and it can be used for different diseases. And I don't have any disease that is my favorite. And as a matter of fact, the blood-brain barrier work I described, it started focus on Alzheimer's because there is indication that the blood-brain barrier gets damaged in the case of neurodegenerative diseases. We have been able to create a disease blood-brain barrier model where the typical proteins that are seen in Alzheimer's patients, like A-beta, are being accumulated and produced, and see how they are able to travel outside of the blood-brain barrier or not, to kind of cleave them from accumulating and tangling before they become too big and they cannot be cleave and remove. So it has applications. So the short answer is I started the blood-brain barrier project with focus on Alzheimer's. And we hope that we can understand better, what the disease is and see if through the soluble materials that come out of the blood-brain barrier in Alzheimer's patients, if we are able to diagnose it at least early.
Yeah, it's fascinating. So thinking about this, there's a question about sensors. What's the best way to mark brainwave irregularities?
Can you repeat, please?
Sure. So someone is asking about the sensors, and they want to know the best way to mark, I imagine, brainwave irregularities.
I don't know the answer to that. I just know that all the data that we collect, the one in the lab, it's very controlled because it's a model and we can test it and it's very reproducible. We have not gotten yet to measure brain waves on patients, and I would, for my understanding and what we are seeing in data that has been collected by others, there is a lot of noise and a lot of variability. So we are going to have to deal with that. In terms of sensor, it's not only the brain waves, that we are measuring. We are measuring a lot of different, from molecules to looking for things, for example, patterns on where you are or how much you are traveling, or even looking at question of sociability. Your phone, detects the Bluetooth of neighbors. And if those neighbors are in your contact list, I can know if you are at home or you are in the subway. If you are in the subway, it will detect Bluetooth, but they are unknown people. So we are trying to use the phone sensors to find patterns and markers that can give us some clues about sociability and the relation of sociability and mental health, mental health disorders. So it's not only brain waves, and it's not only EKGs, heart waves. It's not only that. These are the traditional ones, and of course, it's kind of our standard, but it's not only that we are measuring.
Yeah, so someone was asking that about the phone. So the phone, you're not looking to measure just the blood pressure or temperature. You're actually looking to understand the sociability and how that social life affects one's anxiety and depression. Is that correct?
That's complete. It's totally correct. Yeah.
Excellent. So another question is there a specific drug or chemical substance that could change the morphology of the brain vessels in a permanent or semi-permanent way, i.e. Psychedelic drugs or?
I don't know that this is the case in a permanent way. I don't know, but I think if you have, my understanding is that if you abuse those drugs over and over again, the damage is going to be there. But I don't have the data about psychedelic drugs in my knowledge.
So someone asked, based on what could be observable in your lab, are degenerative brain diseases unavoidable with age, i.e. direct correlation between aging and the appearance of degenerative diseases? Making you work today, sorry.
No, this is I'm thinking, I don't want to have a quick answer. Can degenerative diseases be prevented? It's difficult because we like to put things in buckets and characterize, and say this is a neurodegenerative disease, this is vascular disease, this is so dementia due to vasculature or dementia due to Alzheimer's, neurodegeneration. And that's the traditional classification that doctors use because doctors and everybody enjoys to put things in buckets, and say this is this is that. And by looking in my lab at the blood-brain barrier, that is actually super nice because it's at the interface between what brain and vasculature is not white and black, it's a blur scenario. Can we prevent degeneration? Aging is, this is another part of the, another conversation. We are talking about aging, no? We are talking about, it's a part of aging. So I think degeneration, I don't want to go into immortal, getting us until 200, but degeneration is unavoidable. That's, and there are people with family history and with the conditions they live in, that degeneration occurs quicker. And that's what, based on what we are doing in my lab, is basically subject brain cells from different to different conditions to see what conditions that are environmental conditions, like I mentioned, pressure, friction, torsion, how this can accelerate the damage on the blood-brain barrier or which ones preserve it. So that's what I can tell you based on what I see in the lab.
So if there are no other questions, I'm just going to ask a general question and see if there are others. But one of my big questions, and something that we really like the office about your study, is really the difference between subjectivity and objectivity, right? So you're looking at the cell level, whereas depression and anxiety is also very objective, right? Some people either say they're more depressed, less depressed, and the questionnaire that you gave also has to do with cultural difference of where people are in terms of how they react to it. But ultimately, do you think the cells will be the ones to tell us whether you're really depressed or you're not depressed? Because again, with using the phone, right? Today, there's some faculty at MIT that read your face with AI to decide whether you're happy or unhappy or depressed or how you're reacting. So you're obviously going for the nano cell level. So what's your feeling? What's the big picture on all of this?
It's a fascinating field because it's you mentioned subjectivities. We are all subjective, and it's funny how, especially when we deal with some mental disorders, or not, you don't even have to have mental disorders. You go to the doctor and they ask you, how many glasses of wine do you drink? And you say 1. And they multiply immediately by X, okay, by 4. So it's not that you are trying to lie, it's that our perception of how many you can forget, you don't know. You don't know how many texts have you sent, how many screen. Every time the screen tells me you have spent 3 hours on a screen, average this week, I'm like, no way, so we we have these perceptions, you know, and that's totally subjective, and it's not you being a bad person or lying, is that we have no clue what's going on. So it is very interesting to bring some more objective measures, but without losing the sight. And that's what I'm so happy to be working so close with clinicians, that at the very end is a human being. You know, the technology can tell you whatever, you are dealing with a person. So, and the cultural aspect, again, is something very important to us, because there is a very important database in the UK, for example, in cardiovascular disease, that they have been, you know, sequencing a lot of people. And the algorithm that has been developed with that database has been trained, you know, based on those data points. And Dr. Krieger, my collaborator in Brazil, I just learned from him that that algorithm that is based on the UK database is not being able to predict cardiovascular health on the cohort of Brazilian people. And this is because we are different, we have different populations, and you cannot train algorithms only on one population, because it doesn't work, because it's self-instructed. So that collaboration and bringing different cultures, different, you know, my collaborators in Brazil, in Mexico, and here in Boston, it allows for having that diversity and that inclusion, and having data points from a representative of, you know, and be able to triage the population by different parameters. You know, not everybody is this or that. You know, I still remember when you had the good cholesterol and the bad cholesterol, you know, too. And now you have all these different ones. But I think it will be, we are going towards that, towards a personalized care, and putting in, or taking very serious in consideration that we are all wonderful, but we are all different. So it's very critical, the collaboration with LATAM, to bring the Hispanic world into that, so that we are not being extrapolated or inferred by algorithms that don't represent or persons.
Excellent, thank you. So I'm going to ask one last question, and so when you started all of this research, everyone has a goal, right? Says, if I can achieve this, then I'll be thrilled. Or if I get to this point, you know, I'll have achieved all of what I wanted to study, all of the years I put into it. So let's look at 5 years, 10 years down the road. What do you hope all of this research will achieve? What's the end goal? Like, if you could say, wow, you know, I got to the top, I achieved what I wanted. So how do you see this research in 5 years?
My goal, and this is going to surprise you maybe, is not about the research per se. In 5 years, I would like to have to be able to inspire more and more students and faculty who are not from Boston, from all over the world, to join and work towards battling this terrible disease of, you know, anxiety and depression, and work together and inspire the new generation of scientists, especially girls, who can do it. You know, they can do biology and computer science and then go to medical school. We need that. So for me, is that this is more important than the research per se. So
that's awesome.
I'm sorry, but I am not going to tell you. I will be very happy if, yeah, we are going to publish, we are going to advance, we are going to collect data from different cohorts, and we are going to find things to alleviate pain, for sure, in 5 years. But I think it's more about the collaboration and inspiring new people, and help or contribute, at least put a little grain of sand there to remove a stigma on mental health, which is also a big problem, especially with some cultures. My culture, I grew up where nobody was, had nothing, mental health was not existing. So I think that will help.
All right. Well, thank you everyone for being here today. Thank you, Mercedes. Thanks to IMES for all of the great research they do. I'd like to thank Global Programs, Jacqueline Taylor, for helping out with this. And please tune in. Hopefully we'll learn more from Mercedes as the seed funds continue and as people get to fly again. But we definitely look forward to you having more students and helping the next generation of researchers. So be well, everyone. Take care and have a great rest of the afternoon. Thank you.
Thank you.